He kōna e purangi tēnei nā te reo irirangi o Aotearoa.
We have people developing vaccines. We have people looking at real time how plants take up particular elements into their structure. We have people imaging great white shark heads. We have people looking at poison-tipped darts that they found in a box in the back of Melbourne Museum, and they still want to know if they're toxic. They are. We have people bringing Aboriginal rock art. We have volcanologists. I could go on.
Kia ora, nau mai haramai ki te ao hureha. Hello and welcome to Our Changing World. Ko Concannon tēnei. And it's not an exaggeration. She really could go on.
So our director's favourite trick in a talk is to stand up and say, give me a topic and I'll tell you where we do research on it.
This is Dr. Helen Brand, the Acting Science Operations Manager at the Australian Synchrotron. And with Microscopy Group Manager Dr. Emily Finch, Helen is bringing me upstairs to get my first look at the synchrotron. Wow.
Yeah, it's a good view.
This is a lot.
It is a lot. There's a lot of stainless steel. Yep, welcome to the synchrotron.
Yeah, I'm not often lost for words, but this is one hell of a sight. Spread out before me in this giant warehouse of a building is a labyrinth of shiny pipes, railings, and metal rectangles. It looks like an incredible feat of engineering. You can make out a smaller ring in the centre and then a larger ring around that. And coming off the larger ring, these big blue rectangles, the size of small shipping containers.
This is our particle accelerator, the synchrotron, and it exists to bring us light. So it creates light for us that we then focus and tune to shoot it at samples, scientific samples, to learn more about their chemistry and their structure and lots of different things about them.
I mean, what am I looking at? Which part is it's all synchrotron? Like where are the particles being accelerated?
Synchrotron all the way down. So what we can see here, you can see sort of two main rings. So what we like to affectionately call the synchrotron the science donut sometimes. So what's happening is in the centre there's a gun producing electrons, so an electron gun. Those electrons are being sped up in that inner ring and then shot into this outer ring here where they're travelling at just under the speed of light.
99.9997% the speed of light, to be precise. The storage ring is the electron's final destination and also where the magic happens. Up to this, it's all been about prepping the electrons, reducing them and getting them up to high speed and energy. The storage ring is a stainless steel vacuum chamber. In here, the electrons get steered by bending magnets, and when they're forced to change course by the magnets, they emit energy in the form of light. The light gets collected at different points around the storage ring into what's called a beamline, another metal pipe. And the first section of each beamline has filters, mirrors, and other things to select a particular, shall we say, flavour of light. That's what the big blue rectangles are. Each one of them contains a different beamline and all the equipment needed for research to happen. This is where the scientists come to use the light to investigate whatever it is they're researching. Now, in normal, everyday conversations, when we say light, we generally mean visible light. You know, from like light bulbs or the sun. But when scientists say light, they mean all of the electromagnetic spectrum. This actually goes from radio waves, a personal favourite of mine, all the way up to high-intensity gamma rays, which are very much not good for you. Along the way, you have microwaves, infrared light, visible light, ultraviolet light, and X-rays. In the synchrotron, they produce infrared light all the way up to some gamma rays. But they mostly use X-ray light in many different ways, with a little bit of infrared use thrown in. Now, X-rays, like gamma rays, are also not good for you, which is why the storage ring is enclosed in radiation shielding walls of concrete and lead. and why many of the X-ray beamlines have radiation shielding enclosures and robots to do the work. From our vantage point, I can see the name on the nearest beamline, the Imaging and Medical Beamline. They're investigating new radiotherapy methods for cancer and can do X-ray imaging at high resolutions.
It scans across the sample and then creates it like a 3D model or a 2D model, depending on the science, of what's going on inside or on the surface of or whatever that sample. So yeah, super interesting for that one. And so there's different applications going around the place. There's microscopy, we've got spectroscopy, so squiggly lines that tell you about chemistry and structure and things. Microscopy is more around imaging and mapping. We've got diffraction, which is Helen's specialty.
So we can look at the structure of things like proteins for new vaccines and drug discovery and things like that. And then in the materials space, we can look at how structures of materials change when you do what you want them to do. So in a battery, we can see how the structure of the battery changes as you charge and discharge it. So you can work out if it's going to be a good battery or if it's going to be, you can use it once and then the structure completely falls apart. So it might be really good for long-term storage, but it's not going to be good in your phone when you need to keep charging it day after day, that kind of thing.
And I mean, this isn't the only way to make these kinds of light. So why do it this way? And why do it all in one big giant shed?
That's a good question. So there are lab examples of X-ray machines. They tend to be smaller, so we are brighter than them. So we tend to have more flux density. So we have more light per area that you're going to look at. We're faster, and we have a more stable beam sometimes. Because of the high flux of the beam, we can do faster reactions than you can do in a lab. So in a lab, for example, I'll go back to my technique because that's the one I can easily kind of contrast to. In the lab, a measurement might take an hour. It might take 10 hours. I can do that same measurement here with probably 4 or 5 times the resolution on the data in about 2 minutes.
Okay.
Faster and better.
It's faster and better.
Yeah.
And you may as well, and doing it all together is, you would never build a whole synchrotron for one beamline. So synchrotrons tend to have lots and lots of beamlines. So we don't have many beamlines for a typical synchrotron. So we have 14 at the moment. We're building 4 new ones. We started off with 10, but the ESRF, which is the European synchrotron, is much bigger than us and has 40, 50 beamlines, something like that. We have more holes in the wall that you could put beamlines on, but we don't have the funding to put beamlines on them.
Should we go now?
Yeah, so where are we off to now?
We're going to go take a look at the beamline, so where the science happens. So first of all, we might go to the X-ray fluorescence microscopy beamline.
And microscopy, so this is your domain?
It is, as of last week though, so lower your expectations.
Okay, well, it looks like there's some nice fluorescent drawings down there, so that looks exciting.
They make the best pictures.
Oh, okay.
Yeah.
Okay. Is it a contest?
Sometimes.
So I feel like I'm in a university tunnel of offices and labs, and I can see lots of research posters, very classic department kind of style. But yeah, we're still in that giant building. When I look up, I can see the stainless steel, but in this corridor, it has a very researchy feel.
Yeah, our particular brand of art happens to be research posters, I guess. It's really our MO here. Yeah, so this is the X-ray fluorescence microscopy beamline, or XFM. And so the thing that this beamline does that is really cool is it scans over your samples and tells you where the elements are in your sample, so you can map out things that you can't see with your eyes. So for example, some New Zealand researchers brought in some baby lobsters, and because they just don't know what the key elements are that make up lobsters, like what do they need in their diet to be a healthy lobster. And so what they did is they popped the lobster into the beamline here and scanned over it to see where the elements are. So you can see Around the sides there's all this calcium, so that's in their shell. They need lots of calcium for a healthy shell. They also have arsenic in their eyes and in their sort of main body bit, and copper, it really focuses in their gills. So they used this information to say, okay, well now if we want to develop a diet for lobsters, we know that we need to have calcium, arsenic and copper to make sure that they turn into healthy lobsters.
Some other work involving the University of Auckland, a university from Argentina, and NASA looked at samples from both recent and ancient hot springs to check for elements that were signatures of microbial life. They were testing to see how long these signatures stuck around, so that if we ever got some samples from Mars back down to Earth, we would know how and what to look for to find signs of life. Which is pretty cool. But not Emily's favourite. She pulls me over to a different poster.
Some folks from the National Gallery of Victoria brought in this famous Degas painting, Portrait of a Woman, and they, for some reason, suspected there might be something underneath that image, the famous image that we know of this portrait. So they popped it in the beam, and so the beam's able to scan over the portrait without damaging it whatsoever. And by imaging the different elements in the portrait, they were able to pick up different pigments, which are made up of different elements, and pick out this hidden image underneath the main portrait. And so you can see they've mapped in calcium, in chromium, manganese, iron, to pick up the different features of that hidden painting, just from the pigments that they've used to create it.
That's
really
incredible.
cool. I actually like the underneath one better than the original, the over the top one. This is nicer.
Yeah, and they actually know who this woman is as well. So she was a model that he'd painted several times before, so they were able to identify her. And the theory is that he didn't want to do, the other one was a commission that he didn't want to do, and so he painted over, used an old canvas that he just painted over with lead paint, and then did the commission on top.
So yeah, they can really investigate a huge range of things here. And because New Zealand, through university, research institution and government funding support, has essentially bought into the synchrotron, researchers can apply to come here and do their experiments. We're walking along a corridor on the outside of that bigger ring, so moving in a circle from beamline to beamline. Each setup is slightly different. For the higher energy X-ray light, the beamline itself is behind that radiation shielding. And then next to that, there are offices where the researchers sit and keep an eye on everything. Each beamline also comes with its own dedicated beamline scientists, the people who know what's what, and work with the visiting researchers to help them set up their experiments and maximise their time here. When we head to the macromolecular crystallography beamline, that person is Dr. Rosie Young. Now, micro means small, you probably know that, so macro means large. Though in science, you have to remember everything is relative. Rosie explains what large means in this context.
Big stuff in crystals, which is often like proteins and things which have lots and lots of atoms in them, relative to what we call small molecule crystallography, which is things that are like smaller on a molecular scale. So something like sugar would be a small molecule crystal, but something like an insulin crystal or a DNA crystal would be a macromolecular crystal.
And so what kind of things get looked at this beamline?
All sorts. So yeah, anything from a mineral, it could be a salt, it could be a new drug, it could be a new nanomaterial, to anywhere from proteins, parts of viruses, parts of bacteria, parts of your human immune system, and what their structure is and how they interact with different drugs and different targets. So anytime when we want to know the atomic or molecular structure of something, we can work it out on this beamline. All you need is a crystal, which is the difficult part often.
Did you ever grow crystals at home? With like salt or sugar or borax? Crystals are made when you get this nice repeating shape, over and over. So that's what you need to do with whatever you want to figure out the structure of. Grow it into a crystal. Why? Well, as Rosie explains, the distance between two atoms is too small to see with visible light. even if you had the most powerful microscope in the world. Just 'cause physics. So what they use to figure out the structure is called X-ray diffraction. The X-ray light bounces off the atoms and changes direction. Because they're no longer travelling in straight lines, the X-ray light waves now interact in different ways. Sometimes they cancel each other out. Sometimes they kind of combine to become brighter. And this is what the scientists then detect. But you can't do it with like a few molecules of protein. You need to have the nice repeating pattern of the crystal to amplify the signal.
The crystal is just there to be able to have those like millions of copies of the same thing, to be able to get enough signal in our diffraction to be able to measure it. And we are, with stronger and stronger sources of X-rays, we're able to get smaller and smaller crystals. So here is the second crystallography beamline at the Australian synchrotron. Next door was the first one. With that, we had to have crystals a couple of 100 microns in size. This one, we can have crystals down to about 10 microns in size. And the new beamline that we're just building next door, MX3, will have crystals down to less than a micron in size.
And a micron is 1/1,000,000th of a metre. Human hair is about 60-ish microns in diameter. So teeny. In this office space, sitting beside Rosie, are 2 New Zealand researchers, ready to go with their samples.
Kia ora, my name's Ben Crinkle. I'm a PhD student at the University of Auckland.
And what are you trying to look at?
So with respect to using the synchrotron, we've generated some protein crystals in the lab. For myself, it's a human malic enzyme, which is upregulated in cancer's altered metabolism. And through the binding of ligands to these crystals, we're investigating, looking effectively to develop new anti-cancer drugs.
Okay, and in advance of coming here, what were the steps that you had to do?
Yes, that is probably the hard bit. So we start off by producing recombinant protein in bacteria.
I'll just jump in here. This is a really routine technique used in labs where you use genetic engineering to get bacteria to make heaps of the protein that you're interested in studying. So the bacteria churn out the protein, and then Ben would have done lots of different steps to separate out the protein.
We eventually purify the human protein away from the bacteria. So then we've got a purified human protein sample, and then we undergo protein crystallization trials.
And that is the often very tricky step, right? Is trying to get that protein to form a nice crystal that you can then bring here and shine the light on to figure out the structure.
Most definitely. We put down potentially, so our trays have 96 wells, so that's 96 different chemical conditions, and on average I'd probably be putting down up to 15 to 16 trays before coming for this trip, and that requires an enormous amount of protein to be generated beforehand as well.
And so you've travelled over from Auckland with the protein with you?
Yep. So we've brought refrigerated dewars over with us, so myself and Shay have brought one each, so yes, that comes over in our checked-in luggage.
And how many samples of it do you have to investigate?
On this trip in particular, I have quite a few more samples than some of my colleagues. I've brought 6 pucks of crystals, so 16 crystals per puck, so 6 times 16 is almost 100 samples I've got for this trip.
And what are you hoping that you'll be able to figure out?
So in particular for this trip, we have a new compound that we're investigating, where we have seen through enzyme assays that we have done back home that we're getting very noticeable inhibition with these compounds.
So when you say compound, you're talking about something that might in the future become a drug, because, and when you say inhibition, it's like it's stopping this enzyme from working, basically.
Exactly. Through the use of the beamline here in Melbourne, we're looking to see where it binds. Through the assays that we've done back home, we already know how tightly it binds. This is predominantly to see where in this quite large molecular system the molecule is binding to our enzyme.
Now in his 3rd year of his PhD, it's Ben's 4th time to the synchrotron. With him this time is his colleague, Shay Chunketh.
My name's Shay. I'm also a PhD candidate at the University of Auckland, and I'm, like Ben, using structural biology techniques to study a completely different thing. I'm looking at peptides that are involved could potentially be antibiotics. So my project is looking for new possible antibiotic compounds.
And when you say peptides, you mean very tiny bits of protein, right?
Exactly right. So they're kind of like proteins, but much, much smaller. They're generally made of amino acids. In my case, my peptides that I'm interested in are modified amino acids. So they are similar to the 20 amino acids that make up all proteins, but they have some interesting modifications, sometimes involving things like cyclization. So we can have a peptide, which is usually just a straight chain, like a noodle, but then it can be cyclized or circularized on itself, and that gives it a defined structure.
And have you made these in the lab?
So I've used some computational techniques to find them using a number of different online tools, and then we've produced them in bacteria, much like how Ben explained, recombinantly in our lab using E. coli, which is a workhorse bacteria. And we have attempted to make some of these peptides in our lab, and then we go through a purification process, and we're here at the synchrotron to try and analyze them and see what they might look like in terms of their structure.
So you haven't tested these yet in terms of antibiotic efficiency, but by the computation, that kind of gives an indication that they should have antibiotic properties?
That's right. So we don't know what the actual targets of these peptides might be. They have features that suggest that they might have antimicrobial activity, but we don't know this because we haven't done this testing yet. At this stage, we're interested in characterizing what they look like, and then further down the line, we'll assess that potential function by screening these peptides against a number of different bacteria that we can grow in the lab. Some of them will be trying to test against clinically relevant bacteria, such as MRSA, and other pathogenic bacteria that are important for humans.
And how many have you brought with you to test here?
This time we're focused on just 2 different peptides. One of them is from the soil, actually, a bacteria that is found in the root systems of many different soil environments. We're interested in seeing what this might do. It's made by a bacteria that is known to kill off many different other bacteria. So effectively, it's a weapon used by one bacteria to attack other bacteria and compete.
Is it true to say that essentially after the end of this, you would hope to get like the equivalent of an image, a picture of this really teeny tiny thing that we cannot see, but you'll get a picture of what it looks like?
That's right. We'll be able to hopefully generate a 3-dimensional structure, so we can kind of see what it looks like in terms of its shape, and this might give us some clues as to how it has its effects on whatever bacteria it might have an effect on.
And have you been to the synchrotron before?
I've been some years ago, before COVID, so some time ago, and things have changed here quite a bit, which is really cool. We actually had a tour earlier this morning of some of the new beamlines that have been set up, and it's really exciting to hear some of the capabilities that have been developed over time.
To gain access to a specific beamline, the researchers have to put forward a proposal of what they hope to do and apply for beam time. Ben and Shay don't actually have all that long, and they're not just dealing with their own samples. Here's Ben.
Our beam time is 24 hours, so usually that means we'll be able to slot in as many other biologists as we can. So I think we've got samples from about 6 or 7 other users, so us 2 and 5 back home as well.
Oh, that's a big responsibility.
Yeah, it is. You definitely don't want to be the one who accidentally leaves them at the airport or is responsible for them disappearing. So,
yeah, but I guess you're probably the pro, right, of the group. Having been, is it fourth time?
Yeah, I have done it a few times now, so it definitely does increase the confidence a bit. But it's fair to say you're still a bit nervous when you're sitting in the tarmac, going, "I hope that Dewar that we've got has been loaded on board the plane."
The synchrotron runs 24/6, so they have a long night ahead of them. But each experiment only takes about 30 seconds, says Rosie, so they should have enough time to get everything done. This user room, where they'll be hanging out, has a desk lined with computers and a whole bunch of monitors. Here's Rosie.
So we have a lot of screens here, and we have to have the screens because we can't actually be inside the beamline while X-rays are there, so we have to have cameras to be able to see what's going on. So up there on the top left-hand corner is a big bucket of liquid nitrogen, which is where all of the crystals are kept. So a lot of protein crystals are quite fragile. They don't like being out in the air for very long because they lose all the water in them and they crack and they become a not very nice crystal anymore. And also we perform the experiments at 100 Kelvin, which is negative 196 Celsius, I think. Very cold. And that is because atoms wiggle around a lot with temperature, so they have thermal vibrations, and the colder you get, the lower those vibrations, so they move around less, and so we get crisper pictures basically. So that's why they're kept super cold. So that's that big bucket of liquid nitrogen. We have a robot that comes in and picks up the samples from the liquid nitrogen. The samples are already mounted on what we call pins, and at the top of the pin is a loop made out of either nylon or a type of plastic material, and in the middle of that loop is a crystal. And these crystals are very small. They can be anywhere from 10 microns to 200 microns, and that's about maybe a tenth of a human hair width, so you can't even see them with the naked eye. And that's what we can also see on some of our cameras here. We're seeing the crystal there. So that's the kind of lump on the end of the loop.
And while you were talking, I could see there was a bit of movement in the robot, but you haven't touched anything.
Yeah,
What's going on?
That's because our users can actually access this remotely and collect the data remotely. So at the moment, some of the colleagues of the users here are in New Zealand, and they're looking at basically exactly the same screens on their laptops or on their home computers, and they're controlling the robots. They're able to put in their settings, and they're able to collect their data from wherever in the world you might happen to be.
So you were saying that the beamline can process things really fast.
Yes, and a lot of that's down to our detector, which brought us from about 15-minute data collections about 5 years ago to now 30-second data collections. And that is funded in part due to all the cancer research we do here. So it's funded by the Australian Cancer Research Foundation and also the New Zealand Synchrotron Research Group to help develop those new cancer drugs.
So a lot of cancer research comes through here.
Yeah, a lot of cancer research.
Because it helps, like what Ben is doing, it helps them to figure out how a drug is interacting with a specific protein that's important for different types of cancer.
Yes, and it also in a lot of different stages of the process of trying to fight cancer. So everywhere from trying to understand how a cancer cell works or how the disease is progressing in a human, to testing these new drugs, screening lots of new drugs, to making drugs better. So kind of anywhere along the process, we're doing experiments to try and work out different parts of that.
So that's an intro to the what, how and why of the Australian synchrotron, and a bit of a closer look at the macromolecular crystallography beamline. But we've really only scratched the surface of the New Zealand research that happens here. Next week, we'll be back with another team who are using a different beamline to investigate quantum materials. But for now, thanks to Dr. Emily Finch, Dr. Helen Brand, Dr. Rosie Young, Ben Crinkle, and Shay Chunketh. My travel to the Australian synchrotron was supported by the New Zealand Synchrotron Group Limited. This episode was produced by me with help from Ellen Rikers and executive producer Liz Garton. It was recorded with the help of Otago Access Radio. Sound engineering was by William Saunders. Tēnā koe I whakarongo mai. Thanks so much for listening. Kia pai te wiki.